we can get all of our panelists to be coming back on. I first want to remind the audience to register at learn.omicslogic.com/learn or /projects for your free learning portal account. And if you forgot earlier, I'd like to encourage you to do that now. I'll put the link in the chat box here so everyone can see it. If I get the setting right, here we go. So you can make sure you get there and click on the register free button. So if you can all do that, that would be great. And we need to find Sarah to bring her back in. So Sarah, if you're out there, if you can come back in, we'd appreciate that. And I think we're ready to roll. So gentlemen and ladies and gentlemen, there we go. There she is. We've got Dr. Kasturi Kannan, Associate Professor at Translational Molecular Pathology that we just heard from. Dr. Sarah Zapico, an Assistant Professor in the Department of Chemistry and Environmental Sciences at NJIT. Mr. Ilya Brody, CEO of PineTech Bio in New Orleans. And Dr. Oskalov, who we heard from earlier, Senior Bioinformatician at the Department of Biology, Lund University, Sweden, and the Science for Life Laboratory. Ladies and gentlemen, the first thing I'm fascinated to know is how and when you decided to get into this broad field, and of course, your current niche. And I'm going to call your name. Ilya, let's start with you.
Thank you. Yeah, so I guess, I came to this field just because I was interested in how data analysis could be applied, more in a more applied way, in more biomedical research kind of application. So I think that's the major role. And we talked about how this is being used, to treat cancer, to study different important things that I think are really much more concerning than, where a lot of things in data science are being, used like marketing or something like that. So it seems like a much more compelling reason to apply these skills to these kinds of data sets.
Cool. Sarah?
I realized, yeah, I was mute. Well, yes, in terms of forensic science, as I said before, I always wanted to be a forensic scientist. So I switched after my PhD. So particularly the application of these technologies is because I see the potential for the field in terms of, for example, in challenging samples, identification of human remains. I'm sure that although they are not as using as everyday needs, but I hope in the future we can use it in a regular basis. So, for example, the phenotyping, it would be really helpful to identify human remains, which I mainly what I do, and other things. And for example, in terms of post-mortem interval as well, we have really a need for technologies, and I think based on our previous research, that is what we need to apply these technologies.
Just an observation before we go on, Grant. I just noticed that, we had people here that dealt from childhood cancer, right? So all the way to kind of post-mortem studies. So it's kind of interesting, but there is a distinct difference, right? So there is applied and then there's research. So maybe as you describe your, how you got to this, maybe you could mention, so I understand, Sarah, you're mostly doing research with this, right? It's not actually being used?
Yes, I am doing research right now. For the moment, it's not applied yet to the field. So with my some research, what I did in age estimation, we were able to apply to 1 or 2 cases. But again, it's difficult because of the legal problems. Basically, and also it depends, for example, when I talk about phenotyping, for example, in New York, it's really difficult to apply in cases to determine, for example, who was the physical characteristics of the perpetrator. So for the victim, it's different, but still you need to ask for permissions, etc. So here also the problem is legal issues in terms of with the genetic genealogy, although I am sure you heard about a lot. you have still to ask for permission to use genetic genealogy, and at the end, again, you need to do the SPRs, the full DNA profile. So yeah, the problem right now is the actual application cases, but slowly we are trying to do it. So yes, it's our main limitations.
Fascinating. Absolutely fascinating. Thank you, Sarah. Nikolai? You're muted.
Oh, is he a co-host or maybe? Yeah.
There we go.
Yes, sorry.
I think there you go.
Sorry, I was trying to unmute myself. Yeah, so I'm originally from physics, and yeah, this field. I guess you mean how I moved to this field, broadly speaking, to life sciences. I met medical doctors, clinicians who showed me their data, and I got fascinated by the amount of open questions. Basically, compared to physics, I would say you feel in life sciences and biology, in biomedicine, you feel like a child in a candy shop. There are very many questions. In physics, you kind of have a stable knowledge accumulated over many ages. Basically, in life sciences, it feels differently. There is very little well-established knowledge, which is good for researchers because it's we are curious. We want to know something new, but maybe it's not good from the point of view of patients, because we would like to have more knowledge, of course, and help them.
Okay, thank you. Thank you much. Dr. Kanan, we'll come back to you.
Well, I mean, like I said, I was in a medical research institute where I was doing some core microscopy. And then, I've been a computer scientist, so in a biology institute. So I think it was a logical destination for me to do some bioinformatics. And most of the bioinformatics that I did was I learned ad hoc. I did not have any formal training per se. It's like I'm very curious to know and to understand. So most of it is like trying it out and seeing what it is and stuff like that. And initially I have no idea of what the file formats are, even when I was introduced to SAM and BAM files and things like that. So, but most of the stuff is like, for me, is about being curious and trying to figure out, right? I mean, like using computers, I mean, the terminal was at hand, the tools are there. I mean, nothing really stops me from identifying or, discovering stuffs on my own. So, I learned it on the fly, I would say. And it's, yeah.
Very cool. Very cool. We want to touch on careers in bioinformatics and data science. So open this up to the group to discuss, as you will, how bioinformatics has revolutionized research. What skill sets are required for students to excel in these fields, bioinformatics and data science?
Yeah, so maybe I can briefly start. I think what is interesting to see is that there is this diverse range of data types and approaches, also depending on where they are used, right? So as we saw today, maybe in a clinical setting, it is very useful to have that population level view, so databases and finding the connections between clinical and omics data is very important. In other cases, I think that, what was explained by Nikolai is very applicable to the type of data that just is starting to be generated, right? This is not some widely adopted translational research that is being described. These are cutting edge algorithms and methods that still need to be developed and validated, I would say, right? So I think that depending which direction you go, bioinformatics is becoming one of those domains like data science that becomes, it diverges like a tree into many different domains. So knowing where you're going with this, are you going to go to clinical research? Are you going to industry? Completely different story. or are you going into research? And then which area of research, I think, really will determine and help you understand what kinds of skills. But universally speaking, I think we can all agree that, coding, databases, these are going to be your tools of the trade. And so you should excel and know how to do that. And depending on whether you're a developer versus user, that's also going to determine how much of math and statistics and kind of the underlying algorithms you would need to understand and, potentially develop on your own.
Who else would like to jump in on that?
Okay, let me jump in on that if that's okay. So one of the things is that I feel that math and not math, forget mathematics, but statistics and computing, right? Those are the essential skills that I think every high school student needs to learn, because the applications are enormous. So you can get into any field that you want. So fundamentally, if you're learning statistics and programming, whatever type of programming, I think those are critical. That we can build upon biology or bioinformatics for that matter, right? I mean, if we chose to focus our efforts of programming and statistics in biology, it becomes bioinformatics naturally. So those are the critical skills I think any aspiring bioinformatician or should have, or anyone for that matter should have. And the second point that I would like to focus on is actually network science, learning some network science. I mean, we know that the data are intricately connected and the world is getting connected more and more. So learning some network science will go a long way in applying the learned bioinformatics into clinical practice. So I think these are the two main things that probably that people should be focusing on, statistics and programming, and the second is network science.
Interesting. Nicolay or Sarah? Y'all can just stay unmuted if it's easier for you. You don't have to keep muting. It's up to you. Sarah, go ahead if you got your mute button ready.
Okay. Yeah, I wonder sure. So actually, if you ask me my point of view, I was a student for biotech, as you know, so I learned a lot. I mean, again, you can be a developer or a user. So I think depending on what you want to learn or what you want to apply, you can be simply a user. I mean, it's what I am learning from these courses. So yeah, I think the skills again, one of the important things, at least for me, when I choose students, is their interests and willing to learn. At the end, is the most important thing, and you can learn everything if you are interested in the subject. So yeah, not so much helpful, but I think this is the point of a researcher that I am mostly a user in this bioinformatics stuff.
Nicolay?
Yes. So what skills are important? I would say three types of skills: biology, understanding of biology, programming skills, and mathematical background is also good to have. I basically see maybe I would say two types of bioinformaticians. It can be biologists having a wet lab background who learned computing, who learned programming and math to some extent. And there are also computer scientists who come from a different angle. So they can do linear algebra maybe, but they not really understanding cell biology. And both, I think, can be learned. So basically people with mathematical background can learn biology to some extent, and people from biological background, they can learn math to some extent. So I think, but talking about Lund University, I would say we still have a lack of bioinformatics training. So maybe because bioinformatics is still, it's a new discipline, so maybe there will be more bioinformaticians, but we are kind of still there is no formal training in bioinformatics. It's, as I said, it's either biologists who learn math and programming or computer scientists who learn biology, but there is no like a separate education in bioinformatics, unfortunately.
Maybe to just pick up on that, I think what we heard both from Kasturi and from maybe Sarah as well is, not always is it about those technical skills, right? Sometimes it's that curiosity, that need in your project that really brings you to the point of learning and mastering a skill that you need at the moment. And maybe another perspective on this, bioinformatics is a very diverse field. So it has changed significantly. Today, we heard about how it's continuing to change, and we know that the pace of change is just going to increase. So I think that's another component, right? You have to be driven by your ability to be curious, find answers, but also overcome challenges. And that, I think, is the reason why you need the algorithmic perspective as well, is you need to be not only solving challenge like a tutorial that somebody gave you, but really trying to kind of solve the challenge maybe in a new way, thinking out of the box. You're muted, Grant.
Sorry about that. What would you say to a chemist trying to join the trade? That's from chat. Nikolay, since you were already up, let's let you take that one. You start in on that one. What would you say to a chemist coming into the trade?
Yeah, I've seen people with chemical background, with a background in chemistry. I think they have a relatively smooth path to biology wet labs, because they know all the routines and they have similar, I would say, similar standards. I mean, it's not uncommon. I think chemists who start doing protein research, I've seen many people who migrated from chemistry to biology and computational. I think in chemistry there is, I mean, these guys, they have a good education in, I don't know, quantum chemistry. So it means they understand some computer. They do have good skills in. They should, in my understanding, they should have good skills in computational work. So I see no problems for chemists migrating to bioinformatics.
Cool. Well, let's switch over to what job opportunities and internships for students and researchers there might be. And Sarah, I think, did you have a job available or something like that? If you did, we'll start with you and then we'll hit the rest of the group.
Yes, actually, I have a postdoctoral position. In principle, for a bioinformatician, but I am open to biologists, biochemists that are familiar with this data, precisely because we are going, with the different projects I have right now, we are going to work a lot with omics. For the moment, with transcriptomics and metagenomics. So yes, someone who knows how to analyze this data. So it doesn't need to be a bioinformatician, but at least biologist, biochemist who knows how to deal with this data. So yeah, okay. the post, yeah, I think, yeah, it's already posted.
Great. I think Ilya just posted the link, so thank you for that.
Thank you.
More generally, what do you think, guys? Job opportunities and internships for students and researchers?
Well, okay, maybe sure. I can jump in here and just say that I think it's very critical for people, the students, to have opportunities to intern and maybe even for free participate in a project, right? Because many times when you are taught bioinformatics in a very standard way, you don't have the opportunity to see the project as a whole and really know how useful your work is. So I think internships and especially mentorship are critical. So I know that Kasturi, you do a lot of mentorship. I think we also spoke with Nikolay. You do some mentorship as well. I mean, maybe you can add on to this. How does this actually work in your organization? Is there a process? Do you see, where it's very critical? At what stage of that learning process are these? Is it a critical component? Kasturi, do you want to
Yeah. So, one of the things that we are trying to do here is
who can talk analysis, and then it's important for them.
Nikolai, I think you were on mute for a while, and Kasturi started speaking at that time.
So, I'm sorry. Go ahead.
No worries.
Oh, okay. No, I think, one of the key things that I find it challenging, in terms of, as you saw that we are developing this systems framework, which is heavily dependent on bioinformatics. And of course, I'm mentoring high school students as well. As the key thing is that sustained effort is critical, right? On this, learning some bioinformatics. There are opportunities, very good opportunities, especially the research that we are doing is actually translational. It's directly impacting patients' lives in here. And so, there are these challenges, which I say that, it's about like getting to, like you exactly pointed out, right? It's not just about trying to learn, but also navigating the difficulties that are, on the way, right? Having some troubleshooting the errors and persistently doing that. So, I think these are critical skills that are needed for anybody. But then, yeah, we are training people in there, and so yeah, our program is expanding. So, what was a critical key question in this? Whether I think I'm missing the point here. What were you trying to ask?
What the job opportunities and internships for students and researchers might be, either specifically with your organization or generally. As this program expands, obviously there is going to be, as I see the future is going to be more systems oriented, and already there is something called a Breakthrough Cancer Foundation that has announced about $500 million for integrating data and data integration, and these are going to be the key future prospects. So I think if students can get trained on this aspect of bioinformatics, where they could not just learn how to do just the pipeline alone, but how to integrate together to have a meaningful patient-driven systems, then combining with the databases like the graph databases, the Cypher query language. Cypher, the Neo4j has been like, I'm seeing there are lots and lots of opportunities that are coming, not only in the bioinformatics space, but outside, that are requiring this graph databases knowledge. So I think those combinations will go a long way in securing a good, internship and job opportunities for students. Go ahead, Nikolai.
Yeah, so within our organization, the National Bioinformatics Infrastructure Sweden, we have a bioinformatics advisory program. So all PhD students in Sweden and Swedish academic institutions, they can have a bioinformatician, a senior bioinformatician as a mentor. And it's very important. So I found it very important for the PhD students, because very often those guys they come from they don't have a good environment, don't have a critical concentration of people who can talk analysis, and they need to discuss analytical tricks with analysts. And sometimes I see that people are sitting in the clinics, they're embedded into this clinical environment, but they want to do analysis, and then for them talking to other analysts is absolutely critical, and this is what we are trying to help them with this bioinformatics advisory program. So it is very critical to interact with other people who are doing analysis.
Fascinating. That's really cool stuff. That's a nice program. I want to ask how the interdisciplinary fields are integrated with multi-omics, if you guys want to pick that one up.
Well, I was actually surprised to hear about the way that Sarah is using multi-omics. I think so. Here we have three different ways that multi-omics was described, right? Sarah was talking about just using different omics to estimate the same parameter. And then Kasturi was talking about building a database that has those connections between clinical and maybe multi-omics. And then Nikolai was actually talking about integrating them to see the same experiment, but just from different perspectives. And so I think these are very unique, different types of challenges. But I mean, maybe it's a question whether multi-omics is a must. Is it just something that happened to appear, or is it something that is going to be like a replacement of a single omics? I mean, what does everyone think about this?
If I may go answering that, I mean, I think it's a natural thing, right? I mean, whether we have to subscribe to the single, like I said, right? I mean, it's about reductionism versus systems. I mean, reductionism is not going to go. Studying single omics is going to be there. There are lots of things to uncover. For instance, doing single cell sequencing. I mean, this is about on a single cell level, right? I mean, so it's not yet integrated with a bulk population level. So that's there. But then if we want to understand in a very population centric way how we can put together to have understanding, in a holistic fashion, then obviously multi-omics is the way to go. I mean, it's ultimately the difference in the philosophy, whether we are going to go reductionistic or whether we are going to subscribe to systems. I mean, from our 40 years or 60 years of decades of experience, still we have not yet gone to a place where things could be translated to an actionable, where it can benefit a large class of population. So if you ask me, I would place my bets on systems biology. So multi-omics, is a way to go, in terms of understanding and in fact translating the biology to practice.
Anyone else? All right, let's look at some of the strategies. This is the next question that's come up. The strategies to evolve and involve students in bioinformatics and data science. What kind of strategies do we need to approach?
And maybe before we jump into that, sorry, but I see Ryan here has a good question in the chat. What are your thoughts on the interactome and study integrative cell biology? So I think it's a little bit close to what Nikolai was describing in his talk. I don't know if you want to answer that.
Yeah, so by interactome, I think, yes, talking about maybe by interactome, Ryan means sort of network analysis. And the graphs that I was showing, those were graphs where each node was one cell or one sample, one patient. By interactome, people typically mean a different thing. They mean maybe it's proteins. Then the nodes of the graphs, the features, so I think it's not exactly what I was showing, because my graphs they looked a little bit differently, exactly to drive network medicine. Yes, this is what Ryan is asking. I think it's a slightly different focus. How features interact and how they're linked, how they cluster. Basically, as I said in the talk, you have two dimensions in your data matrix, and you can do cluster analysis and look at the links between your statistical observations, patients, samples, or you can do cluster analysis and look at the linkage between your features. I mean, it's two sort of sides. So I'm doing maybe more one thing than the other. So I'm not very much into the interactome, but it's basically, yes, it is another way of looking at the data, and a very interesting way. So
So I think that maybe, since we are coming to a close, I just want to maybe spend a few more minutes. I think Nikolai, mentioned this aspect of, you can look at either, cells, or you can look at
a lot of
how
robustness.
features interact. I think, Nikolai, sorry, I interrupted you because you were on mute again. But you mentioned how the cell biology, right, is important here because you are studying cell biology ultimately by looking at these different aspects. I mean, how far away is that disconnected from then understanding how those different features are linked? Let's say the cell develops into a certain phenotype. There's some sort of a, separation of these different cell types. Do you look at all at what defines that cell type afterwards? I mean, you must be looking at what genes, right, or what methylation patterns. So that there is kind of like a way to go further into understanding how those clusters are formed and what defines them, right?
Yes. So first of all, you can hear me because I think I get muted and unmuted randomly, and it's not me.
I think it's the network. I think your network.
You're good right now. You're good right now.
Okay, I'm sorry. That might be on my side. Yes. So you were asking about looking at the mechanism. So if you see cells clustering, if you see cells clusters, you would like to understand the what biological molecules drive this clustering of the cells, and if you have the molecular information from the cells, yes, you definitely want to interpret this clustering. So it's a little bit like looking inside your machine learning model. So your machine learning model can maybe find these clusters, but then you want to understand why. It's a next natural question, because you want to get a deep understanding, not just see the clusters, but to understand what they mean. Yes, if you have the molecular type of information from the cells, you definitely can look inside the model and understand why. I hope this is what you're asking about.
And I think that, the next part of what he is asking, the network medicine, that's exactly what Kasturi was talking about, actually, sort of, right? So I guess, as we are wrapping this up, how do these things where do they connect? On one side we have, single cell techniques that generate multi-omics data, and we need to go deeper into that data to really understand what does this mean biologically, and all the way to population health level, to try to identify patients that are very different individuals that maybe also will benefit from identifying these unique clusters of cells that might drive metastasis or something like this, right? So, I mean, where do these things converge, I guess?
Okay. Can I answer this question, or do you want me to? Okay. I mean, I think this is. This is where I think like you exactly pointed out, the cell level. You have these two levels. On one hand, you have the population, and on one hand you have this single cell level, right? Obviously, they have to intersect, and the model that probably that will integrate is again going to be a graph model where we can actually integrate the tables, for instance, that has these specifics of cell, like patients who have those specific alterations. I mean, these could be modeled based on the graph databases. I mean, as I see it, it's not going to be such a huge problem. Right now, the problem is that we are like trying to think these networks are totally different and not looking at a database level, but less looking at just how these connections are from a basic network level, right? I mean, even in network, there is a difference between network biology and systems biology, right? That was my initial question, whether like, okay, why don't we call it as network biology? It's still a biology, but why do we have to call it as systems biology unless we have the power to integrate any type of data that we want, that the graph databases provides us with such a technology to do that, right? I mean, it's a very recent technology. It has a basis on wonderful computer science, right? I mean, pointers and stuff like that. So, it can be integrated, but I feel that the integration will go in the direction of graph databases, and it cannot be just thinking in things in isolation, if you ask me.
Anyone else want to pick that up?
Okay.
Well, not seeing. Oh, go ahead.
Okay. Sorry. No, because of the integration of technologies, I know this is in a completely different field. You know, one of the things we are looking into is the integration and combination of omics for post-mortem interval estimation. So it's kind of what I think you are meaning in terms of health, but in the application of forensic science. So I think also it could be the integration of the different omics could help in that.
Nicolai, any closing comments?
Yeah, I understood Leah's question a little bit differently. He was asking
go ahead.
Population versus single cell, and I know I heard this opinion that typically when we work with single cell data, it's just one individual maybe, and then how does it generalize on the population? And I think it's still early days. We will eventually, hopefully, reach this state that we can sort of utilize millions of cells from many individuals. Sort of the question of generalization of what you discover on one particular sample. It is valid. So maybe all the conclusions might not be generalizable for the population, but still, those are early days. So we just want to see the heterogeneity between cells, and usually it's consistent between individuals. At least the types of cells. I'm talking about maybe blood cells, they are more or less consistent. You see the same cell types from one individual to another. But indeed, there can be diseases where a certain cell type looks strange, and this is what we want to catch. But the more we sequence, the more data we have, the easier it will be to answer a question.
Very good. Well, I'm going to go ahead and close us up. We're at our ending time, and I'm going to put my closing in the chat box also. We want to make sure that we thank our honored speakers today, Dr. Oskolov, Dr. Julia Panov, who was on earlier, Sarah Zapico, Dr. Lisa Taraneko, who was on earlier, Louis Sakamoto, and Ilya Brodsky. So we want to thank you all for being here today and for contributing to this fantastic session. Be sure to join us tomorrow for our second day of learning, thinking, and being challenged by our amazing speakers. And one last reminder, and it's in the chat box, to sign up now at the OmicsLogic sign up page. You click the register for free button, get your free account, and start learning. Thank you all again. We'll see you tomorrow. And yes, the session was recorded and will be available to those who registered. Thanks again. See you tomorrow.